We will just record it for the college website and social media platforms. But if you do not want to make an appearance there, please just switch it off, and that's fine. We will have questions in the end of the talk. So please submit your questions throughout the talk. So at any point that you want, really, in the chat. And I will collate those questions and ask Dr. Taylor then on your behalf. I'm very delighted to introduce Jessica Taylor today. So Dr. Taylor is a Postdoctoral Research Associate in the Gilbertson Lab at CRUK and Cambridge. And she recently has completed her PhD studies, and she did work on repurposing chemotherapeutics to treat Glioblastoma Multiforme at the University of Manchester. And now she's working in developmental paediatric brain tumours and focuses on novel therapeutics that will mitigate post-therapeutic morbidity in brain cancer survivors. And she recently also got a grant supported by the Little Princess Trust, working on how to make the life of children with medulloblastoma easier or help them during their chemo. And with this, I'm handing over to Dr. Jessica Taylor, and we're very curious to hear what she's going to tell us, and welcome her with me.
Thank you, Ava. I'll just get this screen sharing thing for you, and hopefully that will work. Oh, no, that's reading view. I don't want reading view. Okay. Can people see the right thing?
I do think so. Yep.
Yeah, you can see the
looks
big screen thing, and yes, not the little one that I can see as well.
No, with beautiful purple.
Amazing, thank you very much. Okay, so yeah, as Ava said, I'm a Postdoctoral Research Associate at the Gilbertson Lab. I'm also affiliated with Lucy Cavendish, and I'm working on generally reducing treatment-related toxicity for children with Medulloblastoma. So I'll just first give you a bit of an overview of what Medulloblastoma is, and then take you through the kind of work that we're doing in the lab and how we hope to reduce treatment-related toxicity. So what's Medulloblastoma? So medulloblastoma is the most common malignant childhood brain tumour. It accounts for about 20% of all childhood brain tumours, and it occurs most often in children younger than 16. It's classified into 4 different subgroups based on their molecular distinctions. So there's a WNT medulloblastoma, sonic hedgehog, group 3 and group 4. Unfortunately, I think they got bored of the interesting names after the sonic hedgehog one. The standard of care for children aged 3 and over in all medulloblastomas is maximal possible surgical resection of the tumour, followed by radiation to the head and spine and concurrent chemotherapy. So in infants and young children under 3, the radiation therapy isn't used because there's a much higher risk of long-term neurocognitive deficiencies. In terms of the subgroups, we've got about 10% of them are WNT subtype. And in this WNT subtype, they have a very high rate of survival, which is over 90%. So why WNT medulloblastoma? It's rarely metastatic at diagnosis, and the prognosis for children is excellent. You can see from the graph here that with 5-year survival, we've got greater than 95%. So why bother working on the cancer when there's so many other brain tumours out there that have really bleak survival statistics? So despite the excellent prognosis, these children have really substantial side effects from the therapy that they receive. There's a paper from 2017 that has described the long-term effects of treatment in a cohort of patient survivors of medulloblastoma in the US. And these have been compared with their age-matched siblings. At 30 years post-diagnosis, 70.6% of survivors were still alive. And the survivors were shown to be at an increased risk of late onset hearing loss, stroke, seizure, poor balance, tinnitus, and cataracts. And as you can see on the graph here, survivors have a much higher incidence of socioeconomic issues. They're less likely than their siblings to go on to higher education, to marry, or to live independently, or be in full-time employment. They also have a higher risk of learning and memory issues. So what we want to do with our work is to find a novel way of treating the cancer with the same success rate as the 95% 5-year survival, but without all of these accompanying life-limiting side effects. We want to balance that graph, and we want to allow the survivors to live healthy and full lives post-treatment. So how are we going to do this? So targeted therapy is the basis of precision medicine, and it works by targeting specific genes, proteins, or the tissue environment that contributes to the cancer cell growth and survival. By exploiting the fundamental changes that occur in the cancer cells, we can actually maximize the therapeutic index. That's the amount, the window we have of being curative, by improving the efficacy of the anti-cancer treatment, but also reducing the toxicities surrounding the non-cancerous host cells. Cell surface changes in cancer cells, there's many different types. There's loss of contact inhibition, which means they like to grow over each other, which is normal. They have a lot of proliferation and they invade surrounding tissues. Some obvious changes that can occur in cancer cells are a loss of feedback control between the nucleus and the surface. There's increased transfer of nutrients, and there's also the modification and overexpression of specific proteins and receptors on the cell surface. A lot of cancer cells overexpress cell surface receptors. The best known example is HER2, and that's overexpressed on breast cancer cells. And there's a monoclonal antibody called Herceptin that blocks these receptors, and it actually stops the cells from dividing. We want to find a target that's specific to WNT-Medulloblastoma like HER2. But to do that, we need to really understand what receptors are important for WNT tumor cell survival. So what do we know about WNT-Medulloblastoma? So in addition to helping survivors of WNT-Medulloblastoma, we want to increase our knowledge of the WNT tumors and understand the reasons of why they're so responsive to therapy. And we can then exploit that knowledge to build tools to help us make other aggressive brain tumors more vulnerable. And what we know so far, we've learned from establishing a WNT subgroup genetically modified mouse model. This has the same mutations that the WNT tumor has, which are, you can see here, I've got a little pointer somewhere. There we go, that's better. Can you see that? So we've got the CTNMB1 mutation, which is in beta-catenin, and a P53, which is a tumor suppressor, and that's been cut out. We also have another model where the PIK3CA mutation occurs, and that accelerates the tumor growth. So we have the BCP model is quite low penetrance, but the tumor immunoprofile and the transcriptome match the human WNT-Medulloblastomas. And in the PIK model, it's an accelerated and much more high penetrance model, which we developed, the lab developed in 2012. So these mouse models will help us understand how WNT tumors act in vivo and how they are developing slowly over time. We can also use the tumors to implant to do preclinical studies on. We also know from our work with WNT tumors that the tumors are highly hemorrhagic. So central nervous system hemorrhaging can actually result from aberrant angiogenesis and different vascular problems, as well as something, if there's something wrong with the blood-brain barrier. So what we wanted to understand was how these vascular features in different human medulloblastoma subtypes could be of interest, and why the WNT ones specifically are so hemorrhagic. And what the lab discovered was that the secretions of inhibitory factors, which were driven by the mutation in the beta-catenin, it actually induced a really leaky blood-brain barrier. And that permits the accumulation of high levels of tumoral chemotherapy, which could explain why the high survival rates are seen in the clinic after chemotherapy. In our mouse models of WNT-Medulloblastoma, you can see in the image here that we've injected, before killing the mouse, we've injected it with a high molecular weight dextran that's conjugated to a fluorescent probe. And dextran normally can't cross an intact blood-brain barrier. However, when we see here, we see this leakage of the dextran. That wouldn't happen in any of the other medulloblastoma subtypes, just the WNT one. And you can also see here that these fenestrations and the dextran leaking into it on a molecular level means that there's a lot of aberrant vasculature in the WNT subgroup. The normal human blood-brain barrier basically stops anything toxic from getting into the brain. The brain is highly immune privileged, and things like antibodies and other molecules wouldn't necessarily be able to get in a usual basis, which, knowing that the blood-brain barrier is leaky in Wnt-Medulloblastoma, that means that we have the potential to use therapeutics that wouldn't normally be successful in brain tumours. So we also know that we've hypothesized in the past the cell of origin for Wnt-Medulloblastoma. So they, a while ago, the Gilbertson Lab found that progenitor cells from the lower rhombic lip in the embryonic dorsal brainstem are the cell of origin for Wnt-Medulloblastoma, but the precise actual cell lineage was really difficult to demonstrate because there's shared expression of cell markers in lower rhombic lip and pre-lower rhombic lip mossy fiber neurons and climbing fiber neurons. So, but recently a paper by Jessa et al. Confirmed that the Wnt-Medulloblastoma tumours actually mirror the cells that are within the lower rhombic lip. And that confirms that we know that these cells, something going wrong with these cells is causing the Wnt-Medulloblastoma to form in the children. So to be able to study these cells in more detail, what we do is we actually microdissect, as you can see in this little video here. And these are all the different stages of microdissecting from the embryo. And basically you take the tiny little section of the brain and we can culture those cells, we can study those cells with lots of different ways. We can do single cell RNA sequencing on them, we can look at the proteome, and what we want to do is map the surface of these cells and compare them from different subtypes and also normal cell types. And that'll give us an overview of cell surface receptors to determine which are unique and essential for the Wnt-Medulloblastoma development and survival. So here's a big mishmash of data just to give an overview of the kind of multi-omic approach that we're taking. So we have on the left over here, we have some bulk RNA sequencing that we did with our tumour model and tumour cells, just to see what was upregulated at the RNA level. And so you can see here that you've got FZD10, Frizzled10, which is overexpressed in both models. We've also got things like WIF1, and WIF1 is actually one of those secretions that cause the blood-brain barrier to be leaky. The single RNA-seq down here shows that differential populations exist in the Wnt model lower rhombic lip as opposed to the wild-type mouse lower rhombic lip, and shows that there's lots of different kind of cells that we get from doing that microdissection. And here we've got this surface membrane proteomics, which we're doing in collaboration with Paul Lerner's lab. And we've got, what's really interesting for me is that we see that actually the things that we see in the RNA sequencing aren't necessarily translated to the protein level. So we've got WIF1 here, which we know is secreted. So we know there's proteins being made. And we really see nicely here that WIF1 is quite high in abundance in protein, as well as being high in your genetics, in your RNA screens. Whereas Frizzled10 isn't actually highly abundant on the protein, on the surface proteins, but it's still raised over here. So we know that we can't just rely on the RNA sequencing to find a unique target. So the proteomics really is very important. And once we've got all of the different models collected, we're trying to get lots of replicates so we can really get a full overview of different protein expression on the surface. And when we've got all of that together, we can find a unique target to develop our novel therapy. So what is our novel therapy? So we're trying to develop an antibody-drug conjugate. So similar to how HER2 works, where it sticks to the antibody will attach itself to the HER2 receptor and stop it from doing what it's supposed to. Our antibody will hopefully target the unique receptor that we find from our proteomic screening. But what we also want to do is conjugate or attach a cytotoxic agent or drug to this antibody. And that will target the cells. We can use something at a lot lower concentration, because it will only specifically target those cells that are highly expressing this target. Antibody-drug conjugates need to have a really high target specificity and affinity, but then when they work really well because they allow for prolonged drug exposure at the tumour site. We want minimal cross-reactivity with healthy tissues, and we don't want them to cause any immunoproblems. We'll be working in collaboration with AZ to make these, and the expertise available there we'll be able to screen for a really good antibody that has very good target specificity, and develop really good linkers that will enable the internalization and release of the drug in the target cells. And just a little quick explanation of how these work. We've got basically what happens if an antibody joins at the receptor. So you've got your antibody-drug conjugate. The drug is a little star, by the way. So you've got your antibody, which is the little Y thing, and then it binds to the overexpressed receptor on the cancer cell surface. That then gets internalized, and then we have an endosome, which sort of surrounds the antibody and the drug together, has a look at it, sees what's going on. Then it's like, now I'm going to recycle this. Don't need to use this straight away. And it forms a late endosome, which then attaches to a lysosome. And you're going to hear me talk about lysosomes a lot now. But in this case, we'll briefly go past the lysosome. And once it's in the lysosome, the pH of the lysosome is much more acidic than inside the cell. And that actually causes that linker to break off the drug. And the active drug will be released into the cytosol. And then that kills the cell. So what we want to do with this is my main project at the Gilbertson Lab, and I want to create a therapeutic option that has the same survival benefit as the current standard of care. But we really want to reduce the harmful side effects. So using the antibody-drug conjugate therapy, it gives us loads of opportunities to increase the therapeutic index, prolong the half-life, and we've got a number of potential targets that we can see already from our preliminary data. But we do need to identify very unique, very strong targets that are essential to Wnt tumor survival. And this takes a while. As well as identifying the tumor targets, making the antibody-drug conjugate therapy, and then putting it into mice, testing it in mice, and then taking it into the clinic is quite a long process. And I'm impatient and I want to help kids faster. So what we're going to do whilst we're waiting for me to discover these unique targets is another project. So I've recently been awarded a grant funded by the Little Princess Trust in partnership with the Children's Cancer and Leukemia Group. And I'm really excited about this because it's actually helping me to finish off some work that I did in my PhD that I wasn't able to take forward. And the proposed work is aiming to specifically hijack the self-destruct mechanisms of cancer cells and leaving the healthy brains intact. So what I'm saying about repurposing existing drugs to make things go so much faster is that we can immediately kind of skip all of those toxicology tests that we'll have to do to see with the antibody-drug conjugate how toxic it might be, how immunogenic it might be, whereas repositioning, we already know what a lot of these drugs do. How this works is cancer cell lysosomes have been found to be more fragile than healthy lysosomes. And the fragility of these lysosome membranes allow us to use certain drugs to overload it and cause it to burst and release the contents and the acidic contents into the cell. And that actually tricks the cell into thinking that something's wrong with the cell, and it'll start its own self-destruct natural death mechanisms. There's a large number of clinically approved non-toxic drugs that have these effects, and they're called phiasmas. And these phiasma drugs include many antihistamines. As you can see from this little video, these little moving dots are lysosomes. So what exactly is a lysosome? I'm sure you're all on the edge of your seat. So do you remember this kind of diagram from school? This is an animal cell. And then I want you to help me fill it in. I'm looking at you, Naomi. We went to the same school. You were always in biology class. So, what's this thing over here? Anyone? Come on, shout out the back. Can people unmute themselves? Over.
The endoplasmatic reticulum.
Go on, Christina, nice one. Yes. Endoplasmic reticulum. Okay, next one. What's this thing around the cell?
Membrane.
I'm only doing this so I can have a drink of water. It's like I'm going to be really thirsty at this point, so I'll make people do some work for me. This is what I do to my students all the time. Next one. What's this big thing over here? Christina, you're the only one playing.
Cytoplasm.
Nucleus.
Nucleus, yes. Cytoplasm's already labeled.
Yeah.
Finally, we have the most important part of the cell. Can anyone guess what it is? Awesome.
Oh yeah, cool.
So the lysosomes are very important, but obviously not on this graph to be drawn apart from little round things, although that's kind of what they look like. This is a really simplistic view of what happens, similar to what I showed you with the antibody. Lysosomes, the little sacs filled with enzymes and acids, they recycle and remove the waste inside the cell. So here we can see another way that the lysosome fuses with the endosome and degrades. Anything that's useful gets reused by the cell. The lysosome is way more complicated than that. Those little tiny round things that you see, and it's got loads of functions, and it has a specific set of luminal and integral membrane and peripheral associated proteins. The lumen contains all these acidic hydrolases that degrade loads of different substrates, and the enzyme activators and protective factors aid in the degradation. Ooh, sorry. The acidic pH of the lysosomal lumen, the inside of it, is maintained by lots of different processes that are and things that are embedded in the membrane. And this membrane is also has all these lysosome associated membrane proteins called LAMPs. They protect the membrane from degradation, because it's really important. What you need to remember about lysosomes is that all of this stuff inside the lysosome is destructive. If it gets out, the cell thinks something's wrong and it's going to kill itself, basically. So that membrane has to be super strong. And luckily for us, cancer cells, it's not. So basically, cancer cells have been shown to be really similar to patients with NeumannPick. And NeumannPick is a lysosomal storage disease, which is characterized by lysosomal fragility and something called sphingomyelin accumulation. So overactivation of oncogenes in cancer cells actually increases the expression of cathepsins, which decreases those LAMP proteins that were on the membrane. And that alters the homeostasis of the lysosome and causes this fragility. These phiasma drugs phiasma is functional inhibitor of acid sphingomyelinase, which you don't need to remember, but basically the phiasma drugs are weak bases, and they accumulate in acidic compartments. They love the lysosome because in acidic pHs, they become protonated. Due to the positive charge after protonation, they can't cross the membrane again. So they basically get trapped in this acidic environment. And the lipophilic part of the phiasma drug gets anchored into the lysosomal membrane here. So this gets basically protonated and stuck, and that alters the electrostatic properties of the inside of the membrane. So basically the adherence, this is adherent to the edge, this ASMase, and then you get an inhibition of ASMase and you accumulate the ASM. When you get this massive accumulation of ASM, it basically bursts the lysosome, and you get all of those things that we didn't want in a cell into the cell. And because we all know phiasmas are, so here's a really typical phiasma, loratadine. It's sold under the name Claritin. I'm pretty sure most of you will have taken it. I'm pretty sure most of you have intact lysosomes. So we know loratadine doesn't do this to normal lysosomes. It only is very, very specific for cancer cell lysosomes, which is brilliant, because we don't want every time anyone's got hay fever, we don't want our lysosomes leaking all over this place. So you can see here that I've done some preliminary work, and when you add loratadine to cancer cells, this is a normal, cell basically. So you've got your this is normal. This is just DMSO. Sorry, I'm trying to figure out which one have I put in. I put this one in. The little red bits are the lysosomes. And when you add loratadine to a cancer cell at the higher concentrations, it basically bursts the lysosome. And that's what you can see, that fuzzy big orange bit around here. The cathepsin B staining is a bit more difficult. It's not the most clearest. Basically, they should be very punctuate little dots when there's just DMSO, which is just the thing you dissolve the loratadine in. And here you can see it's kind of got a bit fuzzy because they're starting to be released into the liquid outside the cells. So basically, we know it works in cancer cells, which is very nice. As well as that, like I was saying, it was actually part of my PhD, which I really wanted to finish. This work that I did was right at the end of my PhD, and I showed that there's a synergistic relationship between chemotherapy and loratadine, and these are little x-rays of mice, and the glowy bit here is actually a brain tumor. So I made a brain tumor that would luminesce. And basically you can see at 6 weeks, the tumours are reasonably large. And when you treat them with just the vehicle, which is what the drug is dissolved in, the tumour gets bigger over 2 weeks. With the loratadine, it also gets bigger. Just loratadine on its own is not great. And then you put afatinib in, which is a chemotherapeutic, and the tumour gets a little bit smaller. But when you work with actually half the amount of afatinib plus that amount of loratadine, you get almost complete abrogation of the tumour after 2 weeks. And you can see here, there's a massive difference, not massive, significant difference when you add afatinib and loratadine together, and you get a synergistic effect. It also shows that it doesn't actually affect healthy cells here. So HCN2s are normal neurons from a human, and they actually really like loratadine, and they go up quite nicely in viability. So it was really nice to show my PhD work and kind of carry that on. As well, we've got a collaborator, we collaborate a lot with people in Canada who work on other pediatric brain tumours, and they showed that in paediatric, which is a children's brain tumour, another rare paediatric brain cancer, they used Neurotriptyline, which is a plasma drug, and they saw exactly the same effects with this lysosome disruption, which is really encouraging. So what the grant from the Little Princess Trust will allow me to do is basically show that we can replicate that data in WNT-Medulloblastoma cells, and we want to explore more fully how loratadine interacts with the lysosomes in our model systems. We want to visualize changes in the membrane using real-time imaging, and we can really understand how these drugs mediate cell death in WNT-Medulloblastoma. And then finally, I'll be adding plasmas to standard of care to compare survival in our PDX models in-house. PDX is patient-derived xenograft. So we actually take tumor cells from patients and put them in the mice. We really want to show that adding a non-toxic and cost-effective approved antihistamine, by using this in the clinic, that we can reduce doses of irradiation and or chemotherapy. So ideally, we would not use the radiotherapy at all, and we would halve the amount of chemotherapy with the addition of plasma in a perfect world. But I'm sure I'll be able to tell you this in a few months, whether it's worked or not. We're incredibly lucky in the Gilbertson Lab to have facilities and expertise of everyone who works in our preclinical mouse hospital, and this allows me to do this work. One of the recently graduated PhD students, Lisa, has performed resections in the cortex, and she's really perfected these. And that's led me to explore how we might do the same in the cerebellum. With some guidance from a postdoc in Canada, as I mentioned from that other lab, I've managed to optimize fluorescent guided cerebellar resection. And as you can see here, you can visualize the tumor whilst you're doing the operation and take it out. And then basically we re-image the mouse after the surgery, and you can see that the amount of radiance, which is proportionate to the size of the tumor, really goes down post-resection, and the mice survive a lot longer after this resection. So that's really exciting. And we also have access to the small animal radiation research platform. It's called the SARP, and Sabrina, a research associate in our lab, is a real expert in using this machine. And with the SARP, we can replicate the cranospinal radiation that the children undergo in the clinic. And that will allow me to reduce slowly the radiotherapy in the mice, and to see whether adding plasma can allow that radiotherapy to come down, and see whether it reduces neurotoxicity. And finally, we have a great team of knowledgeable cancer scientists who are developing therapies and novel administration methods all the time. And with this, we can compare standard chemotherapy regimens to our novel therapies, or added adjuvants such as the repurposed drugs and plasmas. And as you can see here, we have survival from my PhD work, where we've tried lots of different chemotherapy doses, and we can actually do this with the plasma drugs as well, which will be really exciting. And if you want to learn more about our work at the Small Animal Hospital, I would encourage you to look out for the launch of our Brain Tumour Centre of Excellence website soon, which should be launching in the next few weeks. And with that, I want to thank everyone in my lab and my collaborators, the BRU staff and the core facilities, and of course, my new grant funders, Little Princess Trust and CCRG, the CMIR, which are my CIMR, which are the collaborators I mentioned, and the Children's Brain Tumour Centre of Excellence. Thank you very much.
Thank you, Jess. That was amazing, and a really, really nice overview of everything. We have a question. So please, everybody, just submit your questions, and we can also have it this way that you just unmute yourself when there's a gap and you can ask directly. There haven't been so many questions in the chat, but we have one from Christina. Christina, do you want to ask the question yourself, maybe?
Yeah, sure. So Jess, great talk and a great research, super exciting. So yeah, I was wondering if we know what behaves different in the medulloblastomas, the Wnt ones, in adults. So
Adults.
yeah, so why is it that the
beginning into 1 second.
maybe you mentioned it, sorry, I'm not sure if you mentioned.
I've got a graph, that's all. I'm looking for the graph. There's a graph, there we go. Here we go. So this is the Wnt medulloblastoma, and this is the survival curve. So basically 60 months after treatment, the adults start to deteriorate. And the reason this survival is because they would usually recur. Whereas in the pediatric ones, they tend not to. So I'm not an expert in adult medulloblastoma. My previous supervisor, Martin McCabe in Manchester, works at the Christie, and he actually is one of the leaders in adult medulloblastoma. He does a lot of research in that, and he'd be able to tell you much more why it's less survivable in adults. But I would imagine that it's because either they're more metastatic, or the hemorrhages are more serious, or something around that, but I don't know, I'm sorry.
No, great, thank you. Yeah, it just, yeah, who knows, these things happen all
It's
the
not
time,
very
right?
common at all in adults. I mean, it really isn't. It's not something that is, if you even look at the proportion here, it's mostly young children.
Great, thank you.
Mercedes had a question. Do you want to ask it yourself?
Sure, thank you. Nice talk, Jessica, and I also like your hair. I was wondering if you can explain. I was wondering if you can explain the duality conflict between proteomics and RNA sequencing to identify important receptors in WNT.
Sorry, could you repeat that?
Oh yeah, I'm sorry. My fan is on. I'm sorry. Can you explain like the conflict between proteomics and RNA sequencing to identify important receptors?
The conflict.
Yeah, so
okay, so you mean basically sometimes you don't see a high expression in the proteomics, but you would in the RNA sequencing. Is that what you mean?
Yes, yeah? Thank you.
So that's a phenomenon where some highly transcribed genes aren't actually translated into proteins. So you could have a very high, as we showed with the Frizzle-10, you have a very high gene expression. And actually, if you look at the data in a lot more detail, we see that Frizzle-10 is highly, highly expressed in all of our RNA-seq data, which got us really excited, thinking this will be a brilliant target because it's in the human medulloblastoma data that we have for RNA-seq. It's highly expressed at the RNA level in the mouse models at all stages, in the tumor ones, and it's also in the lower rhombic lip. It was super interesting. And then I actually did an experiment where I got an antibody for Frizzle-10 and I put it in some cells and tracked them and to see if they were internalized, and it was very, very low levels of internalization. So even if the Frizzle-10 was overexpressed as a protein on the surface, if it doesn't internalize the antibody, then it wouldn't work as a target. You get some the other way around where you actually get low levels of gene expression, but high protein expression as well. It just depends on what the cell needs.
Got it. Thank you. An awesome talk.
And we have Wendy saying thank you very much, and she's very much looking forward to hearing the outcome.
Thanks,
from
Wendy.
the Little Princess Trust. Are there more questions, everybody? There must be more questions. Lysosome questions.
I love lysosomes. Everyone should love lysosomes.
Everybody should love lysosomes, that's true.
I'm going to make a t-shirt with that on.
Okay, so Ella is asking, what is the most rewarding part of your work?
I think for me, like the toxic therapy thing is so important. I mean, I've seen firsthand like what the therapies that we give people can do to people for a long time afterwards. And I think the way that people think about cancer needs to change. It's more about not necessarily attacking cancer. It comes across very much like it's a war, like it's a battle. And I'm trying, what I'm trying to change people's perception of cancer is, you know, these people need to survive and live a healthy life after that treatment, and not just, we need to think about it in a more positive way. I know it sounds positive, like we're going to beat the cancer, but actually, if you beat the cancer so much that you actually destroy the person, then you're not winning. And I think talking to people about the differences and why I work on basically a curable cancer, a lot of people will say to me, well, why bother? And the best thing for me is being able to explain this and say, well, we're changing it and we want to make nicer treatments. We want to give people a better life.
Derek has another question. Derek, do you want to unmute yourself and ask yourself?
Thank you. Thanks, Jessica. Really interesting. Are there other WNT cancers where this approach might work?
It depends on if we found a WNT signaling pathway target, then potentially yes. But if it's unique to medulloblastoma, then no. If that makes sense. Does that make sense?
That's fine. Thank you. I was just curious. Thank you.
Mm-hmm. So yeah, it totally depends on the target that we find. Hopefully we'll find multiple really interesting ones that all work brilliantly, but that's me being extremely optimistic. At the moment, I'm just hopeful to find one, and then we can start work. But yeah, I think using this information, we'll get such a nice overview, I think, from all of the different work that we're doing of how these WNT tumours develop, that we will definitely be able to use this for other cancers and to be able to exploit all that knowledge to work with more aggressive cancers as well.
That's actually a very good question. So just to follow up on it, I mean, I have no idea about cancers at all, but how are they categorized? Is it just by morphology mostly, or is it because you might have similar pathways?
Medulloblastoma has been molecularly categorized. Oh God, it's really difficult to say. So basically, we see. I took that bit of the slide out. Why? Let me try and find it. So there's the 4 subgroups that I mentioned. We've got the WNT medulloblastoma, so obviously the WNT signaling pathway, and that's why it's called WNT. Sonic hedgehog, same signaling pathway. It's SHH. It's got a jazzy name. So basically we'll look at those tumours. You can see differences in the pathology when you look at the actual tumours, but in the clinic they would be stained for different things, such as nuclear beta-catenin or overexpression of MYC, which is really common in group 3 tumours. Group 4 is tricky. People don't know that much about that yet, but hopefully they're starting to get a lot more knowledge of all of them at the moment. So yeah, they've been characterized in lots of different ways. Obviously not every patient gets the full RNA-seq whole genome stuff, but I mean, it is quite a small amount of people who have this. So we are lucky that we can do a lot with the tissue that we get.
Brilliant. Thanks a lot. Does anybody else have some questions still? So this is your chance.
Yeah, I've got one. It's Derek again. Jessica, one of the challenges always in these areas where you do reprofiling of known drugs is actually getting proof in a clinical setting.
Mm-hmm.
A major driver to do it. I mean, what thoughts have you got about how this may lead to changes in clinical setting and get the clinical trials done, which, you know, we all know that curing cancers in mice happens quite regularly. It's actually curing cancer was much more challenging. So
yeah,
I think a little bit about the challenges of getting this proven in the clinic.
So I think in adult brain tumours, the dogma is quite strong. Changing anything for standard of care is quite difficult. I used to work in GBM in glioblastoma, and I think, you know, temozolomide is king. Everyone gets put on temozolomide for first line therapy, and it's difficult to change, which is why we kind of did try to change recurrent therapy, because at that point people are a bit more open to trying anything. And we were really lucky to work with clinicians in the Christie. 2 of them were my supervisors, so they were very much onto the let's get people on this drug, let's do it. And that kind of, you know, having that collaboration between clinicians and scientists is really useful when you've actually got someone who can look at the data and go, okay, I'll have a go, I'll try this. And I think that's really important. And having Richard, our lab lead, he was a practicing paediatrician, and he knows a lot of people in the hospitals, and that'll give us a really good avenue for discussing things. Also, there's been epidemiological studies of people on antihistamines in Denmark, and they have a much higher, a much lower mortality rate for solid tumours because for some reason in Denmark, everything gets written down. Like they know who's on Claritin, and they know who's on everything, and they have this amazing data that they can just pull from. And some people studying lysosomal mediated therapies in Denmark have shown that it is actually having an effect epidemiologically on people with cancer. So I think we've got some really decent data to hopefully induce change.
That's really interesting, thank you. So a bit like us taking low dose aspirin to protect us in the future, there may be a position to say actually taking low dose antihistamines will provide a benefit with very little impact on your overall health.
Yeah, definitely. I think that's the biggest thing for me as well, is that we are working with children here, and the one problem with Michael Taylor's approach was he was using, I mean, it's a great piece of work, but they're using tricyclic antidepressant for asthmas. And for you to say to a parent, here, put your kid on this antipsychotic or this, you know, really high dose, you know, antidepressant, for example. I mean, Prozac is a for asthma. So you say, here's some Prozac for your kid. They'll be like, hang on a minute, I'm a bit worried about the side effects. You say to them, we're going to give you some antihistamines. It's not that fear of anything really. Everyone's like, okay, we know what antihistamines are. They're really easy to talk about, and everyone knows that they're safe.
Thank you.
Are there more questions? Anybody else? Nope. It sounds like your talk has been extremely clear, so nobody's asking so
That's
many.
fine.
That's
no, I'm
very
really
good.
looking forward to telling everyone the outcome. It should be really exciting, hopefully.
That is very true. We'll have you in this forum again when you when you have more results as well. Yeah, thanks so much. Thank you everybody for attending.
Thank you all.
As well, and for your questions, of course. And yeah, thanks again, Jess. That was brilliant.
Nice evening.
Yeah, bye bye.